Hello, my name is Ariel Watson, and I am a clinical lab analyst in the microbiology division of the State Hygienic Lab. I'm going to take you on a brief tour of our microbiology section in regards to our Pulsenet whole genome sequencing program. Let's go.
Hello class. Our enteric samples come to us from our accessioning department in these blue buckets. And here are a few examples of sample types that we can receive from the clinical labs. So we can receive stool in either Carey Blair, which are usually orange capped enteric preservation media, or any other branded enteric preservation media. It tends to be like in a green cap. And this is stool along with that preservation media. We usually check this from the clinic for accurate patient name and spelling and patient birth date. In addition to stools in preservation, we can receive pour-off from a larger stool bottle, or we can receive rectal swabs in enteric preservation media, or we can receive isolates that either come to us on a plate. This is a McConkey plate here. We also receive blood plates pretty often for our isolates, or we can receive isolates on a slant, and this appears to be an HIA slant, but we also get chocolate agar or blood agar slants as well. When we receive what we call a full enteric culture, which is a stool from a patient, and we don't know what pathogenic organism is causing them to be sick, we set up a variety of plates and enrichment broths here. You can see that 3 of the plates that I have out are all pink, but they do have different selective chemistry in them, so certain type of organisms will go. Like this black plate, for example, is selective for Campylobacter and is grown at a special microaerophilic growing condition. We have different broths here that help gram-negative organisms grow and certain types of gram-negative organisms. Or this here, which is a KOH, is a very acidic broth that will kill most of the bacteria, but will be selective for Yersinia. If we have an idea of what is in the stool, we can set up far fewer plates. This smaller set here with just one broth is ideal for growing enterohemorrhagic E. coli, and just a few plates with an additional broth here if we're growing up salmonella. If we know what an organism is and we're just confirming before we do the whole genome sequencing, we can set up far fewer plates just to make sure that the isolate is pure and that it's growing under the right conditions. The bacteria we grow and identify requires different growing conditions, and so we have a series of different incubators and refrigerators throughout the lab. For example, here we have a 42-degree incubator, and next to it is a special machine called an inoxamat that can hold our growing conditions specifically for certain types of bacteria that we're looking for. For example, microaerophilic conditions or anaerobic conditions. Here we have a 35-degree incubator that's grown with CO2. And here we have a 35-degree incubator that grows at normal oxygen. Right behind it, we have a room temperature incubator, and as room temperature can fluctuate, this holds the plates at an even 24 degrees Celsius. Here's an example of some plates that have been cultured and grown. This here is a E. coli 0157 sample, and you can see that on a standard McConkey plate, the growth is pink, but we do use plates with more selective chemistry in them, such as this McConkey disorbital and CT plate. Don't remember what the acronym for that is. And it is clear on the McConkey sorbitol plate, indicating that this is an 0157 sample. This is an example of salmonella growing on these selective plates. We expect Salmonella to be clear colonies on a McConkey, and to have this distinctive, what we call H2S, which is a black colony on both this Hectone and XLD plate. And you can see the importance of using plates with different chemistry, because on the Hectone, this H2S grew very well, but on the XLD, we only have a few colonies with H2S. We use several techniques to identify the bacteria growing in culture or the isolates that are sent to us from the clinical labs, including but not limited to oxidase testing, biochemical tests, serum agglutination, and identification from our MALDI-TOF machine. We have 2 rooms that we do most of the library prep and assembly for our whole genome sequencing. In these rooms, we have dead air cabinets like this one featured. The other rooms have biosafety cabinets in which we have the blowers turned off, and they're used as a dead air cabinet to keep amplicon for the different steps of our library preparation separate. And you can see we have tools that make our job easier, like vortexers, multi-channel pipettes. Soon we will have a robot liquid handler, this Eppendorf EpiMotion, in use. Thermocyclers, and then we do have repeater pipettes and the Voyager, which is an automatic pipette that we can program different settings into, and at a click of a button it will move the multi-channel pipette to different settings. This prevents a lot of hand strain and saves a lot of time in our library prep. At the State Hygienic Lab, we have a separate room in which we do all of our whole genome sequencing. The room is in the basement to prevent any vibrations from other aspects of the building, like wind, or right now we have construction in our buildings. We monitor the room temperature and humidity. And here in our room, we have 3 of the MySeq instruments from Illumina and 1 of the Illumina NextSeq instruments, which would allow us to run many, many more samples. We have 3 MySeqs because we have multiple programs that utilize these whole genome sequencers, and in our busy summer months, we do utilize all 3 at once to have a good turnaround time for our samples. This is the Bionumerics software, which is the software that we use through the Pulsenet program to calculate a lot of data for our isolates, such as the serotype, antibiotic resistance patterns, relatedness between isolates. There's a lot of really good information at a glance. Our files do go into an initial database called a RefID, and then after we do some calculation and species identification, we go into an organism-specific database, and this database shows our Salmonella database. So you can see we have our unique keys, the accessioning numbers that have been assigned to our sample from the moment that we received it. There's also information like the date the sample was received, the serotype and specific antigen form, and usually there is an antigen form associated with each of the named Salmonella serotypes. In-house information that's useful for us, like the sequencer run that we put these on, numbers that are identified at a national level for the Pulsenet program, such as this WGS and NCBI IDs. We have something called an allele code that demonstrates relatedness at a glance. The more numbers these codes have in common, the more likely they are to be related. And if they are the same within 6 numbers, there is a very good chance that this is a clustering event. All there's also what we call metadata information, such as the county, city, state, gender, and age of the patient. You can see here that we do have several samples that are listed as unknown, and usually this is listed on unknown in our system when the hospital clinic that sent these samples to us did not provide that information on the test request form. The epidemiologists do have access to that data. If there is an outbreak or a cluster, there should be an outbreak code associated with that sample, and that is assigned by the CDC. And occasionally we do have something called a rep ID that's assigned as well, which indicates that that sample is a reoccurring, emerging, or persistent strain. Within bionumerics, we have the ability to select just a few samples and do different comparisons for them. And you can see here that there are things that we can compare, like antibiotic resistance, to some degree. Plasmids can be compared. We usually use this multi-locus sequencing type ability, which will generate the differences between each of the selected isolates, and the output from the software looks like this, and then we do some Excel magic and export the metadata for the epidemiologists to give them that information in a readable and easy to understand way. This is an example of a clustering event. I highlighted the new samples that we received that week in yellow, but you can see that we did have several samples that were sequenced in previous runs that were all related within 0 to 1 allelic differences, suggesting that this is a cluster.